Modern synthetic molecules, like fentanyl and protein toxins, present a specific chemical challenge. They are lethal in microscopic doses, but physically, they are fragile. For decades, forensic laboratories have relied on gas chromatography to separate mixtures. It is an effective technique, but it relies on an extreme variable: intense heat. To turn a liquid sample into a vapor that can be analyzed, a gas chromatography injection port routinely operates at 250 degrees Celsius. When synthetic opioids or polar drug metabolites are exposed to a 250-degree environment, they decompose, char, and break apart. The heat destroys their structural integrity before they can be measured. Forensic science cannot accurately analyze what its own instruments obliterate. To measure these molecules, toxicologists must use a physical approach that avoids thermal destruction. The solution is liquid chromatography mass spectrometry, or LC-MS. By replacing heat with pressurized liquid streams, this technique isolates fragile molecules directly at room temperature. The analytical challenge is tracing a microscopic quantity of a drug within a chaotic biological matrix, like whole blood or urine. To understand how this instrument achieves selectivity, we are going to track a single fentanyl molecule as it moves through the system. We will follow it through 3 distinct environments: a high-pressure liquid stream, an atmospheric spray, and a deep vacuum. The architecture of the LC-MS system is built to manage a specific transition, moving a target molecule from a pressurized liquid matrix into a deep vacuum without relying on destructive heat. The journey begins in the mobile phase. This is an ultra-pure liquid stream composed of water and organic solvents, like acetonitrile, modified with formic acid. Binary pumps force this liquid mixture through the instrument under immense mechanical force, exceeding 15,000 pounds per square inch. This pressurized stream drives our fentanyl molecule into the analytical column. Inside this stainless steel tube are 1.9 micrometer silica beads, chemically bonded with long 18-carbon hydrophobic chains. As the mixture pushes forward, a molecular tug-of-war occurs. Highly polar background molecules from the blood matrix prefer the liquid solvent. They ignore the carbon chains and are rapidly washed out of the column. But nonpolar molecules like fentanyl are lipophilic. They cling to the hydrophobic C18 chains, tangling within them and lagging behind the rest of the fluid. By gradually increasing the organic solvent concentration, we eventually force the drug to let go. This predictable exit point is known as the molecule's specific retention time. Liquid chromatography separates the drug from biological noise based on chemical affinity. However, the isolated molecule remains in a pressurized liquid state. The next requirement is to transfer this liquid sample into the deep vacuum of a mass spectrometer without compromising the vacuum itself. The instrument bridges these two states using Electrospray Ionization, or ESI, an interface that operates at atmospheric pressure. The liquid effluent enters a stainless steel capillary charged with 3,000 to 5,000 volts of electric potential. This forces the liquid to stretch into a Taylor cone, which emits a fine mist of highly charged droplets. A stream of heated nitrogen drying gas sweeps across this mist. As the solvent evaporates, the droplets shrink, and the density of the electrical charge on their surface increases. The droplet eventually reaches the Rayleigh limit. At this point, the electrostatic repulsion on the surface exceeds the liquid's surface tension. The droplet undergoes a Coulomb explosion. It shatters, stripping away the remaining solvent to release the intact molecule into the gas phase. This sequence is an example of soft ionization. Instead of being destroyed by heat, a proton is added to create a positively charged pseudomolecular ion. ESI completes the phase transition. It turns a liquid into a measurable gas phase ion without using the destructive temperatures required by gas chromatography. The ions are now drawn through skimmer cones into the deep vacuum of the Triple Quadrupole mass spectrometer. Inside the vacuum, the ions enter Quadrupole 1, the primary mass filter. It uses tuned radio frequency and direct current voltages applied across 4 metal rods. Quadrupole 1 is tuned so that only the specific, intact fentanyl precursor ion, with a mass-to-charge ratio of 337.2, has a stable trajectory. Background ions from the blood sample wobble off course and crash into the rods. The isolated fentanyl ion enters Quadrupole 2, the collision cell. This chamber is flooded with an inert gas, such as argon. Here, the precursor ion impacts the gas atoms. It absorbs kinetic energy from these collisions until its chemical bonds snap apart. This is collision-induced dissociation. Because molecular structures are governed by predictable physical laws, the fentanyl molecule always fractures along specific fault lines, generating characteristic product ions. These fragments flow into quadrupole 3, a final vacuum filter designed to sort the pieces created in the collision cell. Quadrupole 3 is tuned to allow only one specific, diagnostic product ion, for fentanyl, a mass of 188.1, to reach the electron multiplier detector. This two-stage filtration means a signal is only recorded if the molecule possesses both the correct precursor mass and the correct predicted fragment mass, effectively filtering out background noise that does not match the specific chemical signature. This two-stage vacuum filtration pathway is known as selected reaction monitoring, or SRM. By tracing the exact transition from the 337.2 precursor to the 188.1 product, the instrument isolates the drug's unique chemical signature from nearly all other biological background interference. This chromatogram shows the result of that filtration. We see a flat baseline that spikes into a single sharp peak for the fentanyl product ion. Because the noise is stripped away, this process can detect drugs down to femtogram concentrations in post-mortem samples. This sensitivity is critical for identifying deadly, tightly structured fentanyl analogs that are lethal in trace amounts. The technique relies on three distinct mechanics: high-pressure separation, atmospheric electrospray ionization, and deep vacuum collision. By operating at room temperature and utilizing two stages of mass filtration, LC-MS allows toxicologists to identify substances that were previously invisible to forensic science. It provides a specialized set of molecular scales that operate where traditional methods fail.